Okay, good afternoon. Please take your seats. For those in the room, I want to say thanks for braving the weather, showing up here. We really do appreciate it. And for those online, I want to also thank you for participating. And to everybody, you are going to hear a fantastic talk. I can personally guarantee that. I've listened to Elaine multiple times talk about this passion of hers. The title of our talk today is Tip to Tail. And I've just been told by Elaine that she probably owe Adam Philippe some money because T2T has been put under some kind of, what did you call it? Copyrighted, and so anytime you use that phrase, you're going to have to pay Adam for it, But anyway, on the joking side. But some housekeeping stuff really quickly. For those online, you can send questions, comments to the chat, and Dr. Sarah Hall is going to be monitoring that, and we're going to put those questions together for the Q&A sections after Elaine's 50 or so minutes of talk. And for those in the room, there's a mic for you to ask your questions, after our presentation. And so just as a very brief introduction, and it's a little bit of history about the Trent lecture. Dr. Trent is our very, very first scientific director, and he was followed by Eric Green, and then Dan Kastner, and then myself. So we've had 4 scientific directors. 3 of them are still very, very active on the NIH campus. So that's a little bit of history. And the other part of the history is that Elaine will be the very first NHGRI investigator to give the Trent lecture. We have had other talk from outside NHGRI within the NIH campus, but she will be the very, very first. So again, Elaine, thanks for agreeing to give the talk. So Dr. Elaine Ostrander is our Chief and Distinguished Senior Investigator of the Cancer Genetics and Comparative Genomics branch at NHGRI and head of the section on Comparative Genomics. Her lab studies canine genome and its utility as a system for informing human health and biology. She received her Ph.D. from Oregon Health Sciences University and did postdoctoral training at Harvard and UC Berkeley. Dr. Ostrander has published over 400 research papers and reviews and won several awards, including the Asa Mays Award, which is presented to research investigators who demonstrate meritorious advances in furthering the mission of identifying, characterizing, and treating canine disease and ailments, again, which is consistent with the work you're going to hear about today. Elaine also has received the 2013 Genetic Society of America Medal, and most recently the 2023 Edward Novitsky Prize in 2020. She was also named as the Human Genome Research Institute Mentor of the Year, and she's a AAAS Fellow and was elected to the National Academy of Sciences in 2019. Dr. Ostrander's current work focuses on the identification of cancer susceptibility genes in dogs, the role of genetic variation in canine behaviors, the origin of dog breeds, analysis of isolated populations, and genetic studies on dogs from Chernobyl. Again, you remember the Chernobyl nuclear incident. So without further ado, Elaine.
Well, thank you, everybody, for being here, for braving the weather, and it's especially great to see so many trainees here. So I'm very grateful to have this opportunity to tell you about the work my lab has done both past and present and very, very recently, towards using the dog model to improve our understanding of human health and biology, and today particularly as it pertains to cancer. What I want to do today is take you on a bit of a journey. A couple of things I'm going to pull out of the vault that are sort of early approaches that we used for studying canine cancer and how that informed similar diseases, similar cancers in humans. And then some things that were published last year, and then some hot off the press things that are one slide literally given to me yesterday. So I want to kind of take you on that journey and give you a full picture, answering the question I get asked most often, which is why dogs? And so hopefully at the end of this, you'll feel like you have an understanding of that. Before I do that, I have to get the slides to work. If you'll bear with me, those of you who've heard me speak recently, I do have to do my 101 primer on dogs. And I'll start by reminding you that modern dogs are the most morphologically diverse land mammal on the earth. And you see some nice examples in terms of morphology and behavior on this slide. All dogs are members of the same species, Canis lupus familiaris, and that includes the 350 recognized breeds. It includes wild dogs, it includes village dogs, includes a whole host of dogs that roam the earth. Recently, and you'll see this, there's been an explosion of resources for doing canine genome studies. And here at NHGRI, we are so fortunate that we've been able to be part of developing those and certainly been able to take advantage of those in really terrific ways. And I also always want to remind people, we don't breed any dogs, we don't kennel any dogs, we don't keep any dogs, we don't direct any breeding. We work with owners, with breeders, with veterinarians, with dog lovers, with people like you. Ours is truly a citizen science project. Now, as you probably gathered from Charles, my lab does lots of things, and it's a really fun place to be. Obviously, we're not going to talk about all of these things today, and some are in different stages of maturity, but I am going to talk about genome health. I'm going to talk about the genetics of cancer susceptibility and studying that in a couple of different ways, and I'm going to talk about our studies in Chernobyl. So that's the sort of game plan that we're going to take. So the way this divides up is I want to start by talking about family-based linkage studies. And in this context, we'll be talking about kidney cancer. And then I want to talk about morphology-related studies, incidences where there's a deleterious allele that's piggybacked along with genes that control a particular trait that's important for the breed standard. And to be a member of a breed, your parents have to be members of the breed, your grandparents, and so on and so forth. And you have to meet a very, very specific, a very, very particular set of standards. I'm going to talk about breed-based studies, and in this context, we'll talk about bladder cancer. I'll talk about dogs as sentinels of environmental contamination in terms of our Chernobyl work. And then I want to talk at the end a little bit about approaches that we're doing, including our studies of regions of constraint, so parts of the genome that can't tolerate mutations and what that tells us about cancer, and genomic burden if we want to look over many, many, many generations and see what's happened to a population. So I know everyone who's ever heard me talk has seen this slide, and I think it's because I've shown it in every single talk that I've ever given. But this is work that Heidi Parker has led for several years, and it's just telling us how the different dog breeds relate one to another. So in this particular dendrogram, she started with 160 breeds. She's now done this with over 300 breeds. She analyzed 10 dogs of each breed on 170K SNP chip, and then she's drawn this neighbor joining tree, showing us how the different breeds relate one to another. And for the most part, it makes sense. Down here are retrievers and spaniels, up here are terriers, over here are the bully breeds. And we're ever refining this because we go back to it over and over and over again to develop our hypotheses. And you'll see some examples of that as we go through, particularly when we talk about bladder cancer. So cancer is the #1 killer of dogs. About 45% of dogs over the age of 10 are going to go on to die of cancer. So dogs who haven't been, hit by cars or gotten infectious diseases. This is certainly a big killer for dogs. And there are lots of examples of elevated risk for specific cancers in small numbers of breeds. And so good examples are osteosarcoma. It's actually a rare cancer in humans, but we see it in the Scottish Deerhound, the Irish Wolfhound, the Great Dane, and the Rottweilers. Invasive bladder cancer, which I'll talk about in Scottish and West Highland White Terriers, as well as in the Shetland Sheepdog. Histiocytic sarcoma, we've worked on that. I'm not going to talk about those results, but it occurs in about 25% of Bernese Mountain Dogs and about 20% of Black-coated Retrievers. And all who get the disease will go on to die of it. So in terms of veterinary medicine, this is certainly a biggie. And gastric cancer, which we see in the Belgian breeds, the Tervuren and the Sheepdog, as well as in the Chow Chow. Now, when I tell people I think it's easier to study cancer in dogs than in humans, at first they give me a kind of puzzled look. But if you think about it, it sort of makes sense. So in humans, sample collection is expensive. There's no doubt about that. Families are small, and in the industrialized world, they're getting ever smaller. There's extensive locus heterogeneity, so a lot of different genes contributing to seemingly similar phenotypes. And we also see examples where the same genotype produces a lot of variation in phenotypes, often because genetic background is very complex. And in humans, environmental exposures, they're varied and they're complex. In dogs, we have some ways of getting around those problems. So we have increased power due to breed structure. Most breeds are closed breeding populations. As a result, there are fewer major genes that are behind each trait. And that, of course, makes the problem statistically much easier. Because everybody has the same genetic background, like you see here in these long-haired dachshunds, it becomes easier to look at genetic background effects. And of course, in dogs, the exposures are more uniform in terms of the pet foods they get, in terms of the health care they receive. As a matter of fact, dogs actually undergo extensive medical surveillance. We spend about $70 billion a year on taking care of our pets, which is really quite extraordinary. Okay, so let's dive into these 4 examples. And the first one that I'm going to talk about is called canine hereditary multifocal renal cystadenocarcinoma and nodular dermatofibromatosis, or RCND. So this is something that we see in German Shepherds. It's autosomal dominant. Early diagnosis is by observation of microscopic bilateral multifocal tumors in the kidneys. And you see firm nodules in the skin and in the subcutis. In females, it's not unusual to see uterine leiomyomas. And I was very fortunate that early in my career, a graduate student, Dora Jonas' daughter from Iceland, came to my lab, and she said, I want to study this, and I brought with me DNA samples from a pedigree of dogs who have this. So, what a great idea. I absolutely said yes. Pretty much got rid of it? Yeah. So the first thing that Dora did was we tested the obvious candidates, tuberous sclerosis 1, 2, polycystic kidney disease. We all eliminated those. But we did have some hints because the renal pathology was reminiscent of a rare human disorder called Birt-Hogg-Dubé syndrome. It's actually been worked at here at NIH by Marston Lenahan and Burton Zabar. Now, Ph.D. patients, there's similarities, there's differences. Ph.D. patients lack the leiomyomas. The RCND dogs don't have pneumothorax, which you often see in the human cases. So we did this genome-wide linkage scan using this very large German Shepherd Norwegian dog family. And here's our cool pedigree. So the troublemaker is this dog here, this male here. He got it from his mom. And you can see he jumped the fence and spent time with a number of different ladies. And in each case, he produced relatively large litters of affected and unaffected individuals. So we mapped this using old-fashioned linkage analysis to canine chromosome 7, and that corresponds to human chromosome 17. And then we were able to use the clinical status because this is autosomal dominant and pretty penetrant in order to get to ever smaller and smaller regions. We were also able to take advantage of various aspects of what we knew about the breed. So this was a Norwegian pedigree. There were also some American dogs. We were able to trace their relationships back anywhere from 10 to 28 generations. And all the affected had the same haplotype. And so we knew they all had it for the same reason. And it turns out that the causative gene is folliculin. So the locus was actually first found in dogs. The gene was identified as the same in Birdhog-Dubé. The gene was actually identified first in Birdhog-Dubé. And you can see in dogs, this is in a really, really, really conserved region of the genome as we go from dogs all the way down to yeast. So I won't talk a lot about the cell biology, which has been worked on since then, but transcriptomic and proteomic analyses uncover links between folliculin, cell cycle control, DNA damage response machinery. Now, interestingly, the homozygous state is lethal in dogs. Given the pedigrees we have, we should have some homozygous, and we don't, absolutely none. And so it's probably not surprising that Birdhog-Dubé knockout mice, folliculin knockout mice, die of kidney failure at about 3 weeks. So a really interesting pedigree and an example of what we can find in dogs, but are incredibly difficult to find in humans. And so we have one we're working on now, and whenever these things come to our attention, which is usually through owners and breeders, these are things that we jump on really quickly. So the first way in which dogs can be very helpful uniquely that we rarely have opportunities to explore in humans. All right, so the second vignette I wanted to talk about is morphology in cancer. And this is work that was led by Danielle Cariotti in my lab, who's now at NCI in Steve Chanick's group. And again, this came to our attention because a breeder contacted us. And he said, you know, I have a lot of poodles who have squamous cell carcinoma of the digit. But he said all the affected standard poodles are black. None of my white standard poodles have this. And I thought it was a peculiarity of his own dogs and reached out. And we were able to get a fair amount of samples from dogs who had this, and 100% of them were black. We never identified a white standard poodle who had the disorder. So squamous cell carcinoma of the digit is actually the most common male bed cancer in dogs. We sent slides of all of our cases to colleagues at UCSF and eliminated melanoma as being the cause. It's locally aggressive. They cut the toes off the dogs, and you'll see on dogs who have multiple toes that have been removed. And it's highly breed specific. So if you're a giant schnauzer, the odds ratio for you to get this compared to the average mixed breed dog walking down the street is nearly 23. If you're a Briard, it's 10, and if you're a standard poodle, it's about 6. Now, with the high odds ratio, we maybe should have worked on giant schnauzers, but we're completely intrigued with this coat color situation. All the cases are black, and light-colored standard poodles are absolutely unaffected. So we did what was the first GWAS study for cancer in dogs, and we very quickly identified a locus on canine chromosome 15. Now, you'll see below that we only used 31 cases and 34 controls. These are all black dogs, black standard poodles. And you might say as a human geneticist, gee, that's not really very much, and you're right. But in the dog world, it's not the number of individuals that matter, it's the number of lineages you sample that matters. And so when we collect dogs, we get their 5, 8 generation pedigrees. We pick dogs that share no parents, common grandparents, common great grandparents, and so on and so on. So we can capture the maximum amount of diversity that exists in that breed. The 6 most strongly associated SNPs were contiguous and statistically significant, and that held up when we did permutations. So going back to your days of studying genetics, we then narrowed the interval. This is the 3 recombination interval. This is a 1 recombination interval. We did this using 84 cases, and it got us down to a region of about 28 kilobases. And there's a copy number variant in there. It's about 5.7 kilobases in length, and it's 183 kilobases upstream from KIT ligand. And KIT ligand is wholly in the one recombination interval. So multiple species conservation tell us that the telomeric edge of this copy number variant has elements of enhancer binding sites. ENCODE, ROAD, chromatin state segmentation analysis also predict the presence of a strong enhancer binding site when they've looked in normal epidermal keratinocytes. So with that in mind, you know, we looked at lots of standard poodles, and what we found is in terms of numbers of repeats, dogs could have anywhere from 1 to 5, 1, 2, 3, 4, or 5. And what we find is that the more you have, the higher your risk is. So one model might be that there's an enhancer binding protein and it binds and then ramps up KIT ligand production. So what we don't know that, but what we absolutely know is that if you're a 4-4 or a 4-5, your disease risk is really high. If you're a 4-3, it's moderate, and we've never seen a case in a black standard poodle who's a 3-3. So a really interesting story here. So KIT ligand interacts with MC1R, which is of course important in coat color. And both of these genes are known to play roles in multiple types of cancer. And everything that I've starred here, these are many of our old friends like BRAF. These are things that we know matter in cancer. So what do we think is going on? I mean, why the coat color differential? Well, it has to do with MC1R. So if you're a light-colored standard poodle, so white, apricot, or cream, 100% of them have a knockout mutation in MC1R. If you are a black standard poodle, none of you have that mutation. So we did do a GWAS study between black and light-colored standard poodles, and MC1R was the only significant difference that we found. And yeah, I'll talk about that in a moment. And that was significant with 100,000 for mutations. So it's as though the homozygous mutation, does it protect light standard poodles? Is it something about the black color that you need, or something in that interaction? I don't know the answer to that, but this was really cool because when you're a breeder and you're breeding for a particular coat color, your selection is very, very, very strong. Dusty black doesn't do it. It's that inky jet black that owners really want to see. And so finding something piggybacking along was something we expected to find, and this was the first example that we found of it, and that was certainly very exciting. In the Briard, black, so I should say, the risk allele, which we define as having 4 or 5 copies, occurs in the same frequency in the light and the dark standard poodles. And based on what I told you about MC1R, that's not surprising. In the Briards, we see the cancer in black and white dogs. But MC1R is not important in making light color in Briards. It's due to beta defensive. So it really seems to be something important about MC1R. All right, so that was really fun, and that was cool, and you know, we still get samples in, and we really need to dig into some of these functional studies. But of course, where our heads are frequently at now is doing genome-wide association studies and sequencing, and in that vein. And a lot of good studies have been published. Our lab has certainly made some good contributions, I think, to that. So Jacqueline Evans was a graduate student, a postdoc in the lab. She now has her own lab at Cornell, and she recently put out a paper identifying 15 susceptibility loci in Belgian breed model, Belgian sheepdogs and taverns and all of those, model of gastric cancer. And while she was a postdoc in the lab, she actually worked on identifying, using a multi-omics approach to identify germline regulatory variants in hematopoietic malignancies in primarily black-coated retrievers and in golden retrievers. And other labs have done similar things, terrific studies published by Eleanor Carlson on osteosarcoma and many others. But what I really want to talk about in this context today is bladder cancer. We've done a lot of work on this, and just recently published a paper on this, and I'm really excited about where this is going. I should say our collaborator all the bladder cancer work is Debbie Knapp, veterinarian at Purdue University School of Veterinary Medicine, and that's certainly important to remember. So why do we need a dog model for invasive urothelial cancer, right? Well, frankly, the cause is unknown in about 50% of people who have the disease, and frontline therapy is complex and it's expensive. 50% of patients who undergo cystectomy will subsequently go on to die of metastatic disease. So if the cancer extends through the bladder to the surrounding tissues or the nearby lymph nodes, the 5-year survival rate is actually less than 40%. Initial chemotherapy can be successful, but resistance commonly ensues, and there's really no second-line treatment. So urologists have told me that median survival times have really not changed for 20 years once you get to the invasive metastatic state. Now, in dogs, it's strikingly similar. This is the most common cancer of the bladder in dogs. There's about 40,000 new cases a year. About 50% of those dogs will experience distant metastases, and the median survival is less than 1 year. So really tragic for owners of these pets. Unlike other models where the cancer has to be induced or chemically treated or something, this is naturally occurring. And that's one of the things that's true of all these dog cancers that offers very specific advantages, especially if you're studying risk. It closely mimics human high-grade invasive urothelial cancer in terms of all the biggies: age of onset, pathology, cellular molecular features, metastatic profiles, and certainly treatment response. So in my lab, this work has been led by Heidi Parker, together with Brendan Decker, who was an Oxfam student in my lab, who now has his own situation at Harvard. And one of the things that they found when they were looking at tumors is that 85% of canine IUC tumors have a somatic mutation in BRAF. And this is the V600E, in dogs is 595, but V600E mutation that we see in so many human cancers: melanoma, thyroid cancer, colorectal cancer. And we know it increases activity of the MAP kinase signaling pathway. In humans, you see this V600E mutation in only about 1% of bladder cancers. So a big difference there, despite all the other similarities. But we also know that more than 1/3 of the human tumors have activating mutations in the MAP kinase pathway. So Brennan and Heidi actually developed a urine-based diagnostic, which NIH declined to patent. And so it was patented by a couple of other places, and companies have sprung up. If you're worried about your dog, if you have an at-risk dog or breed of dog, you can certainly go and get your dog's urine tested, much better than the invasive testing. And finally, as with humans, the environmental contributions are a big deal, and and there's a lot of discussion about them. And Debbie just published a paper showing that secondhand smoke increases risk for bladder cancer in dogs. Very controversial, and we'll see where that ends up. So in terms of breeds, this is again a poster child. Scottish Terriers with their odds ratio of 21, and American Eskimo dogs, Shetland Sheepdogs, and West Highland White Terriers, all shown for you up here, have odds ratios between 5 and 6. So I'll say something about Scottish Terriers in the end, but we decided to focus on the Shetland Sheepdog. And these three are all owned by Dana Drager, a staff scientist in the lab, and I'll talk about her work in just a moment. So we did a GWAS. We found this great locus on canine chromosome 13. 3 adjacent SNPs all gave us strong results. After correction for population structure, which we have to do because every breed has a different population structure, different demography, it remains significant. So the question I would ask is, this is a terrific result at 10 to the -7, but can we see a stronger result if we include data from other breeds? Going back to that wheel that Heidi Parker has been ever refining and ever refining. And we did that. And at the top of this slide, you see exactly what you saw before. That was the initial GWAS. But now we've added in data from collies, from Australian Shepherds, and from Border Collies. And our p-value jumps from 10 to the -7th to 10 to the -10th, and you can see this really strong signal here at 44 megabases in exactly the same position. So indeed, this works really well. The more you know about breeds, the better off you are. Here's the signal. We've blown up that region. There's 20 genes in this 2 megabase region, and the one that we're going to focus on in just a couple of slides is Nepal-1. So we did whole genome sequencing at that point in time from 7 cases and 7 controls. We've obviously done a lot more now. There were 2,000 variants within the locus that follow the allele pattern of those most associated 3 SNPs. Now we compared this to whole genome sequence from 1,700 unrelated dogs, other breeds. And this is data from Dog 10K, which is an effort led by 3 of us to sequence eventually 10,000 dog genomes. And we just put the data out on the first 2,000 genomes. So we found 253 variants in the human predicted regulatory regions and 20 within genes. And it's just those within genes I'll tell you about because the lucky winner here was a coding variant in the Nepal-1 gene. Now, this variant is not floating around in dogs. Of the 1,700 dogs, we found 1 dog who had the variant with a heterozygote. We really only see it in the herding clade. And it's a glycine to aspartic acid change in the 6th transmembrane domain. And we investigated it using all of the usual approaches. I think 11 of them Heidi actually ended up applying, and by all of those metrics, it came out as being highly pathogenic and having impact as a cancer driver. We know it's overexpressed in 3 different studies have shown that it's overexpressed in canine bladder tumors, and we know it's overexpressed in bladder cell lines as well. This single mutation increases risk by 8-fold. That's pretty dramatic. It accounts for about 30% of overall risk in the Shetland sheepdog. You just don't look at 30, 40 human individuals and end up with results like this. And it really speaks to the structure of dogs. We know increased expression is associated with poorer survival and getting through the blood vessels in other human cancers like oral squamous cell carcinoma. And the gene itself is involved in ion transport, primarily magnesium, and lots of people have studied functions of this gene. I won't go into that. But one of the things that we wanted to do is see if we could find some of that other 60%. And so Heidi repeated her GWAS, but this time she controlled for Nepal-1 state. So on the top, all the cases and controls are homozygous for Nepal-1, and at the bottom, they're heterozygous for Nepal-1. And bang, you see interesting loci popping up on canine chromosome 28, on canine chromosome 21, and then down here on canine chromosome 4 and on chromosome 9. So this is pretty cool, because it's really a combination of the heterozygote and the homozygous state that increases or decreases risk. So our current thinking goes like this. The Nepal-1 is really the biggie. You see increased risk in the homozygous state. And the locus on chromosome 28, the haplotype we've identified, is also important and contributes to increased risk. And then it's really these other disease-associated variants on these other chromosomes that ratchet that up and ratchet that down, so that in combinations you end up with increased risk, decreased risk, or no change in the risk at all. And overall, these 6 loci account for nearly 75% of disease risk. So again, really highlighting the advantages of studies of a naturally occurring cancer in just 3 breeds of dogs. So one of the things that I think would have come through very strongly now is that breed structure means everything. And Dana Drager is really interested in this question. And so one of the things she's been looking at is the role of genetic bottlenecks and how it influences breed structure. And now that we have so much sequencing, including sequence of historical samples from the early 1900s that our collaborators like Laurent Franz and Gregor Larson have been able to provide us with, we've learned a lot about what bottlenecks are and what they can do. She's provided graphics of Labrador retrievers and Portuguese water dogs, but the critical information is over here in this panel. So What causes bottlenecks? Well, the creation of a breed with a small number of founders, that's about 6% of the time. Bringing in stock from a new country, again, you're closing the number of the breeding stock. That occurs, that accounts for about 5% of bottlenecks. Formation of the breed club with a set of standards, puts some people out of the club, some people into the club. That's about 19%. Recognition by the AKC actually does not do very much, but popular sires are everything. That's really the biggie. About 30% of bottlenecks are caused by popular sires. And let me just give you a couple of examples. So by popular sire, I mean someone who's overrepresented in the gene pool. It's a common ancestor that has the greatest genetic contribution to the 10 highest producing dogs. That's how dog people formally describe popular sires. It's just the highest producing dogs. Number of awards are taken into account, and then number of occurrences in the pedigree database. So you need a lot of pedigree data to do this, and you need the ability to draw massive, many generation pedigrees, which Dana does. So here's an example of the golden retriever. Okay, in here, in purple, the popular sire was born in 1966. In green, the bottleneck occurred between 65 and 74. And then if we look in 1995, we find that 57% of golden retrievers born in 1995 had the popular sire in their 8th-generation pedigree at least once. We found 3 Goldens that were born in 1999, and they had the popular sire in their 8th-generation pedigree at least 50 times. 50 times. And, you know, we've had fun looking at this for lots of breeds that we care about for lots of diseases. Here's the Belgian breed. The sire was born in 1960. The bottleneck was between 69 and 75. And by 1998, nearly every Belgian sheepdog had the popular sire in their 8th-generation pedigree at least once. We found 4 that were born in 1990, and they had the popular sire in their 8th-generation pedigree at least 22 times. So people often say to me, dog breeders, they cross siblings, they inbreed. Not really. They don't have to because of this, because of the prevalence of popular sires. And a breed like golden retrievers that actually had multiple popular sires over time. And so with that in mind, it takes us to our last scenario, good, right on time, which is dogs as sentinels for environmental contamination. And in this context, we want to investigate the genomic effects of ionizing radiation on free roaming dogs. And I'm referring to Chernobyl. So for those of you who are too young, in 1986, the number 4 reactor in Chernobyl nuclear power plant, Ukraine, suffered a catastrophic accident. There were steam explosions, melting of the core, subsequent fires. And by the way, little known secret, this was actually part of the safety test gone wrong that was responsible for this. 117,000 people were evacuated. You can see that here. Everything was left behind, including pets. This is a school. There are actually just lunches still sitting open on the desk. So we had 3 questions, and this is work led by Gabby Spatola, a graduate student in the lab. What's the population structure of the 800 dogs that are living in the reactor? Yes, in the reactor and around the reactor. And Gabby nicely answered that in a Science Advances paper in 2023. What germline mutations or patterns have been acquired to promote survival of dogs in the region for 15 generations? And do nuclear power plant living dogs possess germline mutational signatures that are consistent with ionizing radiation exposure? So the majority of the dose affected what's now called the CEZ, or the Chernobyl exclusion zone, which is 3,000 square kilometers of Ukraine and Belarus. There's a mosaic of radiation levels, with cesium, iodine, and plutonium being the primary contaminants. And just to give you a perspective, the amount of radiation released was 400 times greater than that released at Hiroshima and Nagasaki together. A lot of radiation. And that elevated background remains, and the potential for ongoing damage remains, and that's what we're interested in. This is actually a Chernobyl worker, and you can see that he's holding 2 puppies. The photo was taken just a few weeks after the initial explosion, and there was a push by the authorities to destroy all of those dogs that had been left behind because of concerns that their fur, their paws would carry radiation. But obviously dogs got through, and that's why there are dogs in the region today. So how do we get involved? Well, at the invitation of the Nuclear Power Plant Authority, the Clean Futures Fund, and Tim Rousseau from the University of South Carolina, and he's the key collaborator here and co-mentor of Gabby Spatola, went to Chernobyl. They set up spay-neuter clinics and health clinics at 4 different points in time, 2017, 18, 19, and 2022. They did it both in the power plant, and the power plant's over here in purple, and there's a sort of a spreading of those depending where in the power plant they sampled. And then over here in Chernobyl City, and that's where the workers live, it's about 15 kilometers away. Radiation is much lower here than it is over here. They've taken over 300 blood samples. This came to NHGRI, and most of these actually have now been sequenced by NIST. So the first thing Gabby had to do was figure out their population structure. So you can think naively, if you're looking for something, well, I'll just take the dogs from the power plant and the dogs from Chernobyl City, and I'll sequence them, and I'll do a GWAS, and anything that comes out is something that's important. And that could not be more wrong, right? You really need to know the population structure because all dogs have elements of their historic ancestry and recent admixture. So the historic ancestry is by and large German Shepherds and German Shepherd-related breeds, the Caucasian Shepherd, Alpine breeds, so on. And then they really diverge. Nuclear power plant dogs have very little recent admixture. There's a little like in the Russian Hound, but not much. Whereas those in Chernobyl City have recent admixture with Doberman Pinschers, Schnauzers, Alpine-related breeds, St. Bernard, and the Rottweiler. And that's because the workers in Chernobyl City are obviously bringing their dogs and their pets in, and that's why we're seeing the very different population structure. The thing to keep your eye on is the German Shepherd. So that's coded in aqua. Every column is a dog. Over here is the power plant. Over here is Chernobyl City. And this is giving you information about which breeds are here. And really what you want to look at is the aqua, and you can see the prevalence of the German Shepherd in both of those. So the first question Gabby tackled was an analysis of the current exposure of germline using a family trio analysis. So this is familiar to you, I'm sure. Ionizing radiation is the trigger for oxidative stress, increases mutation rates. Exposure during gametogenesis causes DNA damage passed from parent to offspring. So you see de novo mutations, the offspring, they're not present in the parent. So Gabby and Ruben Buckley, a postdoc in the lab, did a kinship analysis of all the dogs that we had, and they were able to identify many, many close as well as extended families. I think 77% of the 302 individuals in this analysis ended up somewhere on the graph, with the biggest being this data set of over 160 individuals. Purple are nuclear power plant dogs, and green are Chernobyl City dogs. And you can see things like this include dogs from both. Others include dogs from one another. These 3 dogs here are 3 dogs that are actually living in the spent fuel rods. Breeding and living in spent fuel rods, really remarkable. So they looked at these individual pedigrees, most of which were really complex, some of which, as I say, included dogs from both populations. And we ended up identifying 31 trios. 24 from the power plant and 7 from Chernobyl City, and this is all unpublished work from Gabby. And then you need controls, right? So we had 25 control trios, and they included things that we obtained publicly, things that Dog 10K, the large sequencing effort, had produced, and things that we had done specifically here at NHGRI. And there is one caveat, which is that the depth of sequencing differed. So over here, you're looking at Chernobyl. We sequenced all those. The depth ranges from 19 to 35x, whereas because of the different sources here in the controls, there's a much greater range. So do we find de novo single nucleotide variants that differs between Chernobyl City, the power plant, and the controls? No, we do not. And so this is not statistically significant. We don't see evidence in this particular kind of analysis of single nucleotide variants. When you look at the mutation spectrum, transitions are certainly more prevalent than other kinds of mutations, but within the groups of 3, you don't see any significant differences. How does that compare with published results? It's pretty much the same. So here are the 3 things you just looked at, with the control dogs being unexposed. We had access to a very small cohort of German Shepherds. There were only 4, but they had deep sequencing, and their mean number of de novo SNPs per generation was the same. Ditto for a group of 43 dogs sequenced at 40x. These are both unexposed. It's the same. And honestly, that's what you see in humans too. And this is work that's come out of Steve Chanick's lab. So he looked at offspring of cleanup workers. And although you see more de novo mutations than you do in dogs, it's the same if you look at exposed versus unexposed. You really don't see any difference. So we thought about that, and then we decided, we're really not done with this problem. Maybe we haven't been asking the right questions and maybe not in quite the right way. So moving forward, what we really decided we want to focus on is mutational load and fitness. So rather than looking at a particular snapshot or moment in time, we actually want to look at the effects of accumulated exposure over, say, 15 to 25 generations. And here, what you want to know is the difference in the number of mutations on a given IBD segment, identity by descent segment, the same or different in your 3 populations. So the story, as always, starts with that German Shepherd because it's in everything. And over here in blue is a segment of interest, and it's passed to the Chernobyl dogs, a set of controls we collected from Eastern Europe. At a time when this was really very difficult, we had a huge number of generous collaborators who sent us samples under difficult situations. And then here's that German Shepherd control. Time marches on. Let's say the nuclear explosion occurs here. Obviously, you're accumulating mutations as you go on. And then when you get up here, you can look and you can say, well, is it different? Do we see more in the Chernobyl dogs than in the Eastern European dogs? And what is the control telling us? And key to this is identifying these IBD segments, looking at runs of homozygosity, which is work that Gabby is doing now. This is all unpublished. Now, the one thing about this is it doesn't tell you anything about fitness. So you can find these differences. You don't know if they affect fitness, although you can certainly hypothesize that variants that transition to being in the homozygous state are certainly likely to. And so Gabby's also looking at genetic load and constraint and fitness. So remembering that demographic changes will influence the genetic load, the load of deleterious mutations you have. So she gave me this nice visual yesterday. So imagine, okay, so these are our founding population for dogs. These are all wolves. There's an ancestral allele here in tan, and there's a derived allele here in dark brown. And in order to achieve fitness, all of these dogs have at least one copy of the ancestral allele. But the derived allele is certainly starting to pop up, say, 20, 30,000 years ago when dogs were domesticated. Time marches on, things happen like bottlenecks. We now have a much smaller population, and now we see those fitness effects. And again, you have to have one copy of the ancestral allele to achieve fitness at this locus, and if you have 2 copies of the derived allele, then you do not. Now, we know we need to do deeper sequencing. We know we need to look at things besides SMVs. We need to look at indels, tandem repeats, and so on. But I think this has tremendous promise for answering these questions of what's gone on with these dogs in Chernobyl. And this is just the second to the last slide that I'll show you here. So there's a couple ways to think about it, and then there's one little piece of preliminary data that I think leads me to think this is going to work. So Gabby has used a very large cohort, largely the dog 10K dogs, to determine rare variant frequency across the codon sequence. And so evolutionary constraint indicates mutational tolerance in positions with missense or synonymous substitution outcomes. And as I said before, most of the homozygous mutations are deleterious. They're going to be reducing fitness. And so a good example for how to think about this is something that anyone who's studied this has certainly seen this analogy. A fighter plane that comes back from battle, and everywhere there's a red dot is somewhere where a bullet ended up. And so one group of people would say, oh, gee, this is where we should really reinforce the plane underneath all these red spots. But then the engineers say, no, 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 we should reinforce at the places where we have nothing, no shots, no mutations, because these are the planes that didn't make it home. And so the same applies here. So we measure this evolutionary conservation in terms of philo-P scores. And so over here is an individual. He has a very low philo-P. This position has a very low philo-P score, and you can see that there are a lot of mutations that are tolerated at this position. So this is somewhere in the wings, it's not in the engine. Over here, by comparison, this has a very conserved region, a very high philo-P score, and you can see mutations are not well tolerated at all. And so this is absolutely key for everything we're doing. So hot off the press, Gabby looked at this in, again, German Shepherds. And she looked in modern, post-World War II, pre-World War II German Shepherds as well as wolf dogs. So German Shepherds became a breed in 1906, all right? So when she looks at modern German Shepherds, she sees a fair amount of homozygous loads. She goes back in time and she looks post-World War II, it's quite a bit less. And then if she looks at pre-World War II, it's a lot less. And we know events like world wars certainly affect population structure. Either dogs become less popular, people can't own larger dogs, they're difficult to feed, or just the rebuilding of the breed using popular sires often has a big effect as well. And this is nicely validated by the creation of wolf dogs. These are the Sarloos and the Czechoslovakian wolf dog. And they were created by combining a German Shepherd with wolf dogs. And so anything the German Shepherd has at this point in time, that's what you're going to see in the wolf dog, and that is exactly what we see. So that's kind of cool, and it gives me hope this is going to work. And so then yesterday, Gabby maybe hesitantly showed me this because I got maybe too excited about it, but I think it's very cool. So here she's looking at positive selection and genetic load. So selective sweeps decrease heterozygosity in the sweep region, and that can result in an increase in deleterious allele frequency through hitchhiking. And remember, we talked about that. We talked about squamous cell carcinoma of the digit. So she did a scan of the genome. She used non-overlapping windows of 100 kilobases, defined her candidate sweep regions rigorously, and she found 32 of them, which was really awesome. 14 of them are actually on chromosome 3, which is interesting. Don't know what it means yet. Have just was literally yesterday, and so we've just begun to investigate this. But we're encouraged because these are going to be things that are important in being a German Shepherd. But we expect that piggybacking along are going to be those deleterious alleles that have been hard to find just by mapping genes for body size and then looking around nearby. But this is a much more elegant and successful way to do it. And some of the genes under these regions are certainly encouraging in that respect. She sees genes that are, for instance, important in spatial reasoning, genes that are important in hearing, and so lots and lots of work to do there. But boy, am I excited to dig in on this. So I'm going to go ahead and stop there. I just wanted to make a couple comments on the future of dog genomics. Breed-associated cancers clearly offer opportunities to study risk. One of the things I didn't talk about is the integrated canine data commons, ITDC. And this is something that was started and maintained by NCI. People put their data in there, especially clinical trials data, which I haven't talked about at all about being a veterinarian, but other people could get up here and fill hours talking about clinical trials in dogs and how that impacts thinking in humans. And the idea is to further human cancer research by enabling comparative genomics of canine cancers. Studies of breed relationships, the more we know, the better it is. And I didn't put this map up. We're currently sampling literally from all over the world. Evolutionary constraint and mutational burden are key to exploiting the dog as an environmental sentinel. And there's other places in the world where we have samples that we're just beginning to explore. Deep sequencing, we need it, and T2T are needed. And very fortunately for us, Adam Philippi and Jeff Schonenbeck have taken the lead on the canine T2T, canine sequencing, and Miska's doing some deep long-reach sequencing. And that's just so important. Expression and functional studies are critical. It's not somewhere where canines have really led the pack. One Health is a concept that was developed several years ago around infectious disease. And the idea is you don't think of dogs and humans as silos that occasionally crisscross, but you think of them as a very integrated way. And your thinking should really facilitate both veterinary health as well as human health. And the more we facilitate veterinary health, the more that reflects back on human health and human biology. More functional resources are certainly coming. A lot of this is coming out of the Broad, and that's really exciting. If you want access to our data, you can come here and you can scan, and as usual, we're making information available prior to publication, as we did with the Dog 10K project. So I can't name all of my collaborators. I highlighted the people in the lab who led the studies that I've talked about with little stars here. These are people currently in the lab, and these are some of the past contributors without whom these things certainly would not have happened. Just to mention again, Tim Musso at the University of South Carolina on the Chernobyl work, and Debbie Knapp and her team at Purdue in all of the bladder work, and the big pedigree work we did with Frode Ringwith at the Norwegian School of Veterinary Medicine. So I'll stop there. And I hope what I've given you is a flavor for thinking about dogs and humans in a much more integrated way, and understanding how human health can be facilitated by studies in dogs.
People online. Well, it's not a very good question. Yeah. So Elaine, I just genetics of dogs 101. Do they have the same number of genes as humans?
Yes.
How many chromosomes? Because you kept referring to.
I'm sorry. Yeah, I should have said that. So they do have the same genes as humans. For long stretches, the order is the same, but the dog genome is broken up into 38 pairs of chromosomes in addition to the X and the Y. Sorry about that. I should have said it.
My not very good question is, what were all those dogs doing in Chernobyl? Were they just stray dogs that?
Yeah, so we believe they're offspring of the pets that were left behind following the accident. So people literally ran, literally ran, got on buses and just went, and have never been back. And so these are the dogs that weren't destroyed by the authorities who made it through. As you saw, that worker was playing with a couple of those dogs. And at their max, there's about 800, and we have samples from about 400, 300, 400 of them. Nope, don't have any of that information. I mean, it was just such complete chaos. No.
Thank you, great talk. Do we have any hypotheses about why mammalian gametogenesis seems to be so robust in the face of all this radiation exposure? Yeah,
we've certainly thought about that a lot, and we had a lab meeting with Steve's group to think about that. And I don't have any, I'm not an expert on this, so I don't have anything real profound to say other than something to do with redundancy, right? That has to be part of the story somewhere, although I don't know where. And so I guess I'm going to have to defer to people who know about reproductive biology on that one. Sarah.
We have a question online from Binta Jallo, who's interested in what kinds of IRB review processes, but I guess
Sure.
I would call it IAPOC and other oversight processes.
Yeah.
You have to go through specifically when analyzing DNA from dogs owned by the researchers.
Yeah, so what we do, and so my lab used to, about 2/3 of my lab used to work on breast and prostate cancer. So we have this mindset that this is so important. So every time we take a sample, we get a signed consent form. And we actually worked with the IRB on developing the consent. They thought it was overkill, but we thought it was important. It would be important as time marches on. And so, we get consent to get medical records, pedigrees, consent to recontact, all of those things. Every dog is coded. We don't release publicly any personal identifiers, dog's name, ID, owners, any of those sorts of things. And we get consent to do genetic studies and to publish the results of those. And we're not a certified lab, so we don't return individual results. So it's definitely something we've thought a lot about. And even though we don't have to, we go back to IACOC every year and say, anything you want different, anything you want changed. In the case of Chernobyl, or the case of the studies we did in the Galapagos, there's nobody to consent. However, we do have to get permission from the local government authority. And for instance, in New Guinea, when we did the study on New Guinea singing dogs, that was something that actually took a lot of time. So all those things do happen, even when we're looking at free breeding populations.
That was a wonderful talk. I just have a question, more sociological than scientific question. So we all know stories of kind of sharp elbows and a lot of competition to find human disease genes. I'm just curious about in the dog genetics community, similar, different, or how does the community work together on these problems, or is it more independent labs?
Yeah. So I often comment on that, and especially having been in the human breast and prostate community, the dog community works really well together. And the reason, and I'm not being Pollyannish, it's very practical. There's limited funding. And so my success is everybody's success. Eleanor Carlson, the Broad success is everybody's success. The veterinarians at Davis, it's everyone's success. And so it's really one of these situations where everything rises up. And so there are people who, years before there was a human genome project, had dog DNA samples in the freezer. And those people have come forth, and they are full partners. They're not sample providers. They're full partners. And we've all had that kind of mindset. And they're authors on all the papers. And a lot of this is driven by owners. And breeders. They want their dogs healthier, and they don't want to hear any crap about, well, you have to give samples to me, and if you give them to her, I'm not going to work with you. Won't put up with it. And I think Dog 10K, which I alluded to, so this was a big collaboration. NHGRI took a lead in this. It was, 16 different countries, labs across the world. Everybody pulled samples. Everybody contributed analysis time. Nobody had a grant to do this. And I think it really set an even higher standard for how to work and play well together. So I'm proud of the community.
I was curious about something you mentioned at the very start, that a lot of dogs get cancers.
Yeah.
Is that also true of wolves, like say in zoos, that elderly wolves get cancer?
So, right. So even more dogs would get cancer if they live longer, if they weren't hit by cars, which is still the leading cause of death, as I understand it, in dogs. So elderly wolves, I don't know, and I don't know if there's a big enough survey. When I read all these papers about which breeds have which cancers, I never see data. It may exist somewhere, but I don't think that's a big problem. I'll look it up for you. It wouldn't surprise me if it's, kidney disease or something like that. Out in the wild, no way. Survival of the fittest, not a chance, right? And so maybe those alleles have just, they're not there. But let me look it up for you and see what wolves in zoos and in conservation centers tell you. That's a great question.
Thank you for your talk. I had a question about the Chernobyl dogs. And so I saw your student speak at the symposium, and she has said that those dogs don't live as long.
Nope.
Maybe you don't observe as much incidences of cancer or illness, but I just wondered.
Right. So I always get asked the question, how much cancer do the Chernobyl dogs get? We don't know, because it's a very harsh environment. It's cold. There's limited food. And, they live on average to be about 3. And how much of that is genetic? How much is the environment? We don't know that. But one of the things we've talked to Steve Horowitz about is how to do aging studies on those dogs, right? And so we're not there yet, but I think we've got some ideas about how to approach that. So yeah, they don't live a long time, but it's not different as near as we know for dogs in the power plant versus dogs living in Chernobyl City.
Thank you.
Superficially, not any rigorous study. John.
2 questions, both of which are just, I'm having a hard time wrapping my head around what you're finding. One is the poodle toe cancers. How can that not be melanoma?
Yeah, I know.
I mean,
I know.
MC1R.
I assumed when I got it, I said, oh, this is obviously melanoma. And all of that said no, and I said, I don't believe it. And so I sent slides to human melanoma geneticists at UCSF, and they said it's absolutely not melanoma. And so what it is in relation to melanoma, I'm not enough of an expert to know the answer to that. But I mean, I kept beating myself down about exactly this issue. And remember, in other breeds, you do see it in white standard pools, if that makes a difference. But yeah, MC1R, have to have an intact MC1R and the black pools together.
And I do wonder if the constant contact.
You know, I wondered about that. Yeah, I wondered about that too. And that doesn't seem to be the case because you don't see more or less of it in show poodles. So obviously they're being judged on everything, so they're not going to have calloused foot pads than what you see in pets. So I don't think that's it. I don't think it's an environmental stimulation.
Again, second question is, again with the germline, I'm having a hard time jiving that with the observations of the kids from Hiroshima showing all these birth defects for several generations.
Yeah, I mean, I think if you go back and you read that literature, it's not as huge as you would think. The number of birth defects is much lower than lore, and that's something that Steve actually pointed out to us. But remember also, the children are in a much more protected environment. If you're born and you're stillborn and you're a dog, we never know about you. Or if you're born and you have some, you know, dramatic thing where you're missing feet or paws, we never know about you, right? You never come to our attention.
I'm going to do a quick follow-up on Sean's question. I mean, it is a different cell of origin. So you're saying that these are keratinocytes that are having the mutations and the genes are active, and that's interesting, and they don't metastasize then because it's not melanocyte being. So let me check. So the word metastasize, I'm a little squeamish on, because what owners find is if they have one toe affected, the chance of getting another toe affected is higher than just a random standard poodle. And people came to us with, 3 toes cut off from their standard poodles, which of course locked them out of the breeding pool no matter how many shows they worked at. Is that metastases? We never had anyone. Well, so that has not escaped us, actually. The microbiome issue has not escaped us at all. And there are people tentatively trying to do microbiome in dogs, but I think people don't know how to start, honestly. I don't know if that's metastases or not. But for sure, if you've got one, you're more likely to get more. I know, but that's not a melanocyte.
For all I know, it's genetic background. It's some allele and genetic background. Sure. Okay, sure.
All right. From our online attendee. First question is, are the levels of somatic/mosaic mutations the same between the Chernobyl dogs and non-exposed? And or 2, assuming gonadal mosaicism is a driver of apparent de novo mutation, are there differences in the de novo rate on the paternal haplotype versus maternal haplotypes for the Chernobyl dog?
There are not differences in the maternal versus the paternal haplotype. I think Gabby's looked at that. And so what was the first question again? No. So that was what I was showing. So when she looks at levels of de novo mutations in Chernobyl dogs versus free breeding dogs from Eastern Europe, we don't see differences. The mean is the same.
Again,
all right. Thank you.
Say, I hope you have enough ammunition. Now, somebody asked, why are you studying dogs at the NIH that you can adequately and directly answer this question? I want to thank everybody for
Thank you, everybody.